Now let's bring it back to a moment to what was the point of all of that. What is the point of translation? The point is to make proteins, and a protein is a polypeptide, and a polypeptide is amino acids that have been linked together via peptide bonds, which is what we accomplished here. I got a chain of amino acids, an amino acid polymer, polypeptide. And once a polypeptide has been folded into the correct three-dimensional shape, it can become a functional protein, which means that way back when we were studying cells, and I told you the job of ribosomes is to make proteins, hey, we just did it. We just accomplished that task. But it's a lot more complicated than that one-sentence definition of what a ribosome does would lead you to expect. Finally, termination. Termination occurs when we get to one of those 3 stop codons. If you check the chart that we had of all of the different possible codons that exist, the genetic code chart, you'll see that there are 3 that are marked stop codons. The tRNA with the stop codon doesn't have an amino acid attached to it. So when it goes through the ribosome, when it travels from the A site to the P site to the E site, there's no new bond that's formed, and therefore our polypeptide is released into the cell. Now, where does this occur? Well, in the ribosomes, obviously, but where do you find a lot of ribosomes? The rough endoplasmic reticulum. So this polypeptide is now in the rough endoplasmic reticulum, and we can bundle it together, put it into a transport vesicle here, and then we can send it to the Golgi apparatus in order to obtain its final three-dimensional shape and get packaged and sent off to wherever its final location is. So, all of these different processes we've just talked about, DNA replication, transcription, and translation, where are they occurring? Well, for DNA replication, that of course happens in the nucleus. The DNA is not allowed to leave the nucleus. Remember the central dogma. When you're trying to remember this stuff, remember the central dogma. When you use DNA to make more DNA, that's called replication. Since DNA is not allowed to leave the nucleus, any process that involves DNA must take place in the nucleus. So that's true of replication, but also transcription. When you take DNA and you work it into RNA, that's transcription. So transcription happens inside the nucleus. RNA processing takes place inside the nucleus as well. And then only after we get the 5' cap and the 3' poly A tail on that mRNA molecule are we allowed to send it out of the nucleus where it finds a ribosome on the rough endoplasmic reticulum, and there we can translate from RNA finally into proteins. Okay, from RNA into proteins, that's the process that we just talked about, translation. The finished polypeptide is then sent to the Golgi apparatus where we're going to get some folding done. The finished polypeptide is going to change its shape, it's going to snake around, it's going to fold, it's going to twist until you get the final 3D structure of a protein. Right next to me here, floating up and down and twisting around, we have a globular protein. Some enzymes are globular proteins here. So this might be what the final finished product would ultimately look like. Now, proteins obtain that three-dimensional shape spontaneously. It's just the interaction of the different amino acids on that chain kind of playing with each other that determines how it's going to fold and twist and obtain that final shape. But they will do that part mostly on their own. Now, you might not want an unlimited number of a certain protein in your cells at any given time. There are times when the cell has to kind of turn down production of some protein and then turn up production of another one. A very easy way for cells to do this is just to control transcription and translation. For instance, if I took those transcription factors editors, right? And I told them, do not lock on to this promoter region of the DNA molecule. Do not lock on there, then that would prevent any gene from being transcribed. And if it can't be transcribed, it can't be translated. So controlling these mechanisms, those transcription and translation, is how the cell can control its own metabolic functions and its own development. Now, one thing that you should get from all of this is that the original DNA sequence at the very, very beginning is important. It's incredibly important to what that molecule is going to look like at the end. If you change even one base of that DNA molecule, you could change what that ultimate protein looks like. And sometimes this happens. While DNA polymerase is very, very good at its job, it does make errors from time to time. And when it makes a little error, a spelling mistake, we call that a mutation. So mutation is when your genes change, when you have some sort of spelling error in your DNA. It was supposed to say ATT, but now it says ATC. Sometimes mutations are fine. You can pretty much ignore them. They won't do you any harm. Sometimes they are catastrophically bad. They are deadly mutations. Some mutations might affect you very, very slightly. Other mutations might actually be beneficial to you one way or another. So that's what we're going to look at next, is mutation.